The ultimate goal of us in this discussion is to really get a good understanding of the electron transport system. However, first we need to go back and make sure we understand where we started from before we get into the electron transport system. So we're going to go all the way back to our first glucose molecule. When we learned the steps of glycolysis, we learned that we start with a 6-carbon glucose molecule. We go through several steps, ultimately breaking that glucose molecule in half into 2 equivalent molecules, glyceraldehyde 3-phosphate. Each of these glyceraldehyde 3-phosphate molecules will continue down until they both ultimately are converted into pyruvate. Through this process, 2 ATPs are used, but 4 ATPs are produced. So we get a net production of 2 ATPs. During this process, we also produce 2 NADH molecules. All of these steps occur in the cytosol. After the pyruvate is made, the pyruvate is then transported from the cytosol into the mitochondria, where we then move into the citric acid cycle. The citric acid cycle begins with pyruvate being converted into acetyl-CoA. During this reaction, again, we produce an NADH molecule, and we also release one of our carbons as a CO2 waste product. We then enter into the citric acid cycle by acetyl-CoA here, combining with oxaloacetate to produce citrate. Citrate is then mutated or isomerized into isocitrate. Isocitrate then begins the steps of oxidation reactions, where through each step we are releasing NADH molecules. The overall process of the citric acid cycle, one complete turn, produces 3 NADH molecules. One of these steps, in which succinate is converted into fumarate, produces an FADH2 instead of an NADH. The importance of this electron carrier being slightly different will come into play here in just a moment. While we're discussing energy equivalence, we also do, during one step, by breaking a thioester bond, we produce a GTP, which is an equivalent of 1 ATP. All of these steps in the citric acid cycle occur in the mitochondria, actually within the matrix. The matrix is the area within the mitochondrial inner membrane. So if we look at this picture of the mitochondria, this is the outside membrane. The inner membrane here, so the space within the membrane is the matrix. The space between the outer membrane and the inner membrane is called the outer membrane space. Now that we have taken our glucose molecule, we have completely oxidized it, completely broken down the carbon's backbone, extracted all electrons out that we can, we are now ready to move into the electron transport chain. In the electron transport chain, we are going to go through a series of oxidation reduction reactions in which we are going to take our electron and move it through the different parts of the electron transport chain. If we look at this slide here, it demonstrates the different oxidation reduction reactions that occur. As NADH comes in, it is going to first become oxidized itself into NAD, and NADH will give its electron to a flavin mononucleotide molecule that is found within what is called complex 1. Complex 1, the NADH coenzyme Q oxidoreductase, is a protein complex found in the inner membrane of the mitochondria. This protein complex has 2 areas, the FMN and then the iron sulfur area, that have the ability to be reduced and oxidized. So as we go, NADH gives the electron to complex 1. Now NADH is oxidized. The first part of complex 1 is reduced. Then we keep going with the electron, give the electron to the iron sulfur area. Now this becomes reduced, and the first portion is oxidized. Now that the electron is ready to move from complex 1, it must go through a carrier molecule to get to complex 3. The carrier molecule itself is called CoQ, or coenzyme Q. Coenzyme Q will now be reduced because it received the electron. Now coenzyme Q has the ability to transport the electron through a new oxidation reduction reaction to what is known as complex 3. Complex 3 is known as the coenzyme Q cytochrome C oxidative reductase. Similar to what we saw in complex 1, we continue with these oxidation reduction reactions and transport the electron through the molecules. The electron keeps moving through oxidation reduction reactions until we get to our next carrier, which is cytochrome C. Cytochrome C is now reduced because it received the electron from complex 3. Then cytochrome C is going to donate that electron to complex 4, the cytochrome oxidase. Complex 4 is a collection of several different cytochromes that take turns completing oxidation reduction reactions to carry the electron through the complex. At the end of complex 4, the electron is transported, combined with an oxygen, to produce a water molecule. This is a very long, drawn-out process, it seems, but it happens very quickly. This is a way for the NADH molecule to give the electron away. All the NADH was doing was carrying the electron to the ETC. Now that electron moves through. Each successive step of the electron transport chain is an exergonic reaction. We are releasing an energy buildup each time we go through one of these oxidation reduction steps. Notice complex 2 is here at the top. It's at a slightly different place of the inner membrane of the mitochondria. This is why the FADH2 production in the citric acid cycle is important. FADH2, produced as succinate is converted into fumarate, goes into complex 2, the succinate CoQ oxidative reductase. Electron keeps moving through a series of oxidation reduction reactions and eventually moves into coenzyme Q, then can continue in the same manner we observed with the NADH. Since we have electrons coming from two different areas here with the coenzyme Q, we have something called the Q cycle. The Q cycle involves an extra cytochrome that allows an electron to hang out within the Q cycle while we are waiting for the electron to be able to move into complex 3. So at this point, we should understand that the electrons are moving through oxidation reduction reactions as it goes through the electron transport chain. This is technically the electron transport. What we need to think about now is how can we couple this movement, the transport of electrons, to the actual production of energy. The production of energy is known as oxidative phosphorylation. Because we have been using these oxidation reduction reactions, we're going to couple it to the activity of another protein enzyme complex within the inner membrane of the mitochondria known as the ATP synthase or ATPase. As we look at this picture here, it shows you exactly what we were looking at in the last slide with the oxidation reduction reactions, except it is giving you a better idea of how these protein complexes are situated within the inner membrane of the mitochondria. So here comes the NADH, donates the electrons, moves through complex 1. These are oxidation reduction reactions, gives the electron to CoQ. CoQ gives the electron to complex 3. Any extra electrons will cycle through the Q cycle, then eventually go to complex 3, then into the cytochrome C, moving to complex 4 until the electron can ultimately be combined with oxygen to produce water. As each of these steps occur, protons are transported from the matrix into the intermembrane space. So as we see this happen, NADH donates the electron, it moves through. As the electron moves to coenzyme Q, this was an exergonic reaction with a negative delta G, allowing us the energy to move the electron from the bottom to the top, into the intermembrane space. Same thing occurs as we go through the different complexes. The ATPase has this structure We have the F sub-zero region, which is found within the membrane of the inner membrane of the mitochondria. And then we have the F sub-1 portion that has different binding sites for the molecules ADP, phosphate, as well as ATP. The F sub-zero region that's found within the inner membrane of the mitochondria is made up of a series of alpha helices which allow a channel or pore to be formed which can selectively let protons or hydrogen ions flow through the inner membrane of the mitochondria. Okay. So now let's put all of these things together. Electron moves. Okay. And I haven't made this point yet, but it is important to see that when the NADH does release its electron, a proton is also released as well. So we have a whole lot of proteins down here in the matrix. They are there. So as the electron begins to move to CoQ, we pump some protons. Then as the electron moves from CoQ through complex 3 to cytochrome C, pump 4 more electrons. From cytochrome C to complex 4 all the way down to the oxygen, pump 2 more protons. Hopefully you can see that as we continue to move more and more electrons through this electron transport chain, a large amount of protons will be collecting into the inner membrane space. We learned in the beginning of the semester when you have a higher concentration of a molecule such as a proton on one side of a membrane, it can flow by facilitated diffusion through a specific channel down its concentration gradient. It turns out the ATPase will be here so that those protons can flow back through the ATPase down their concentration gradient. Now that the protons are back down here, they can be pumped to the intermembrane space again. If we look at how this is going to work, here's the matrix where all of our NADH is made. Once the electron hops through the electron transport chain in the inner membrane of the mitochondria, protons are moved. Now we have a very high concentration of protons, a proton gradient, in the intermembrane space. Now those protons can flow back through the ATPase down their concentration gradient. As the proton flows through this ATPase, we are going to take an ADP and a phosphate, combine them together to produce energy. How can this function? It turns out that as the protons are flowing through the ATPase itself undergoes several conformational changes. It can exist in 3 different conformations with 3 different binding abilities. In the first conformation, the O or open conformation, it doesn't really like to bind anything. Once it moves into the loose binding conformation, it's going to bind an ADP and a phosphate. So if we look over here on our picture, first it doesn't really want to be bound to anything. But a proton flows through, now it binds ADP and phosphate. Then another proton flows through, changes conformation again, and that ADP and that phosphate are going to be put into close proximity to each other, and the catalytic reaction will occur that combines the ADP and the phosphate. Then proton moves through, another conformational change. Now we are in the T or tight binding conformation. This is the point when ATP is finally going to be released. So you can think of this almost as a motor protein that is turning. When it turns once, it has nothing. Turns again, grabs an ADP and a phosphate. Turns again, puts the ADP and the phosphate again, puts the ADP and the phosphate together, producing ATP. Turns again, now it's back to the doesn't want to bind anything, so it spits out that ATP, and ATP has now been produced. This is the point when it's important for us to make sure we understand the difference between substrate-level phosphorylation and oxidative phosphorylation. We saw ATP being produced in glycolysis as well as the GTP being produced in the citric acid cycle. That was all substrate-level phosphorylation. We used an exergonic reaction and coupled that release of free energy to another reaction that simply combined an ADP and a phosphate to produce ATP. Now what we've been discussing is oxidative phosphorylation. This is where we used the exergonic reactions that were a series of oxidation reductions, moving the electron, producing the proton gradient. The proton gradient then allowed our ATPase to produce our ATP for us. The last thing we need to think about is the yield of ATP we get per glucose molecule when we are completing the electron transport chain. Recall that from glycolysis, we had a net of 2 ATPs and 2 NADH molecules. Experimentally, we can see that for every NADH molecule, we get the equivalent of approximately 2.5 ATPs. Now, how can this possibly make sense? Remember that if we look back at our electron transport chain, as the electron from the NADH is coming through, we don't just get 1 proton. We get a total of 10 protons coming out into the inner membrane space. Those 10 protons flow back through the ATPase. So that allows our ATPase to turn pretty close to 3 times. This is why we say an NADH gives us the equivalent of about 2.5 ATPs. So back to our overall energy yield. In glycolysis, those 2 NADHs times 2.5 gives us an equivalent of 5 ATPs. We then move to the Krebs cycle. In the Krebs cycle, we had 1 GTP produced, which is equivalent to 1 ATP. We then made 3 NADH molecules. We made 4 NADH molecules, 3 in the citric acid cycle itself and 1 in the decarboxylation step in which pyruvate was turned into acetyl-CoA. That gives us the equivalent of 10 ATPs, and we made 1 FADH2. Now, if we look at this picture, we can see that FADH2 does not go through complex 1. FADH2 came in through complex 2 and took the electron directly to coenzyme Q. Therefore, we did not get 4 of our protons pumped. So we don't get quite as many ATP equivalents from an FADH molecule that's carrying our electron. We actually get 1.5 ATPs. So right now, to review, we have made 5 ATPs per molecule of glucose in glycolysis, and we have 12.5 molecules of ATP made by the citric acid cycle. But recall, we make 2 pyruvates in glycolysis. So we go through the citric acid cycle twice. So instead of 25 ATP, 12.5 ATP, we actually have 25 ATP. So we typically say that per glucose molecule, we end up with a total of 32 ATPs. Now there are times when we may not quite get our 32 ATPs. Sometimes we only get 30. And this is because, recall, glycolysis occurs in the cytosol. The NADH molecules from glycolysis are not in the mitochondria. So they have to get in there. Unfortunately, NADH can't just jump straight into the mitochondria itself. So the NADH has to be incorporated into a molecule. One possible shuttle we see is where dihydroxyacetone phosphate reacts to produce glycerol phosphate. This reaction uses NADH. So the electron is actually incorporated into the molecule glycerol phosphate. Glycerol phosphate has the ability to move now into the mitochondria. In the mitochondria, glycerol phosphate can be converted back into dihydroxyacetone phosphate. The mitochondrial phosphate dehydrogenase enzyme is a little bit less efficient, so when we do the reverse reaction in the mitochondria, we only get an FADH2. This is going to overall decrease our energy yield from a total of 32 ATP to 30 ATP. To get the 32 ATPs per glucose molecule, we have to use